In the winter of 1902, a park ranger counted the last wild bison left in Yellowstone. The number he came up with was 23, not 23,000, not 2,300. 23 animals huddled in a remote snowbound valley, all that remained of a species that once blackened the plains in herds of 60 million. What almost nobody knew back then was that those 23 survivors, and the strangers brought in to save them, would leave a genetic fingerprint so specific, so durable, that scientists would still be reading it in blood and bone more than a century later. And what that fingerprint has just revealed is forcing researchers to go back and question things they thought they already understood about how this herd survived, how it was rebuilt, and what it actually is today. This is not a story about bison grazing peacefully in a national park. This is a story about a genetic time capsule buried inside living animals, and what happens when scientists finally crack it open. Every one of the thousands of bison currently roaming Yellowstone today is, in a very literal sense, walking evidence. Their blood carries a signature written before automobiles existed in that park, before airplanes flew overhead, before anyone had ever heard the word DNA. Researchers drawing samples from these animals in the field are not just studying a modern population. They are pulling up a buried record of a rescue mission, one carried out by ranchers and rangers who had no idea their decisions would still be traceable, gene by gene, more than 100 years later. And the deeper researchers dig into that record, the more it becomes clear that the tidy version of this story, the one told in park brochures and nature documentaries, was never quite the whole truth. To understand why this DNA discovery matters, you have to understand just how close the American bison came to vanishing completely. Before European colonization, tens of millions of bison moved across North America in herds so immense that early travelers described them as a moving brown sea stretching to the horizon in every direction. Estimates range from 30 to 60 million animals. Within the span of a single human lifetime, that number collapsed. Commercial hide hunters shipped hundreds of thousands of hides east every year. Railroads cut through the herds and encouraged sport hunters to shoot bison from moving train cars for entertainment, leaving the carcasses to rot. And layered on top of the commercial slaughter was a deliberate campaign. Military and government officials understood that destroying the bison meant destroying the primary food source of Plains nations, and they pursued that outcome as a matter of policy. By the late 1880s, a species that had numbered in the tens of millions had been reduced to a few hundred animals scattered in isolated pockets across the continent. Yellowstone was one of those pockets, and it was a strange kind of accident that it survived at all. The park had been established in 1872, not primarily to protect wildlife, but to preserve geothermal wonders like geysers and hot springs. Bison happened to live there, in the remote high country, and for a while that remoteness was their only protection. It did not last. Poachers pushed into the park's interior despite the risk, because a single bison head or hide could fetch enormous money, at a time when the animals were nearly gone everywhere else. Army units administering the park at the time tried to stop the poaching, but resources were thin and the terrain was brutal. By 1902, the wild, free-ranging population inside Yellowstone had been hunted down to that now famous number. 23 animals tucked into the remote Pelican Valley, the very last wild bison surviving anywhere in the lower 48 states without ever having been captured, fenced, or crossbred with cattle. Here is where the story could have ended, quietly, in extinction. Instead, a strange mix of federal law, private ranching, and improvised conservation kept the population alive. In 1894, Congress passed the Lacey Act, which finally gave park officials real legal teeth to prosecute poachers operating inside Yellowstone. That bought time. But 23 animals is not a population that can rebuild itself on legal protection alone. The genetic math is brutal. A group that small is one hard winter, one disease outbreak, one bad breeding season away from blinking out completely. So in 1902, park managers made a decision that would echo in the bison's DNA for the next 120 years. They went out and bought bison from private herds to supplement the wild survivors. 18 females came from a herd in Montana known as the Pablo Allard herd, animals that had been gathered up from remnants of wild bison years earlier by ranchers, who saw value in preserving the species even as everyone else was hunting it into oblivion. 3 bulls came from a separate ranch herd in Texas, built by a former cattle baron named Charles Goodnight, who had captured some of the last wild calves on the southern plains and raised them privately, driven as much by fascination and opportunity as by any conservation ideal. A handful of calves were also drawn from the surviving indigenous Yellowstone herd itself. These animals, the 18 Montana females, the 3 Texas bulls, and the small number of native-born calves, were brought together and raised at the Lamar Buffalo Ranch in the northern part of the park, largely kept apart from the wild remnant herd that continued to live and roam in the Pelican Valley to the south. For decades, this arrangement was treated in the historical record as a simple, tidy story. 23 wild survivors nearly went extinct. Ranchers and government officials stepped in with 21 more animals. The 2 groups mixed. The population recovered. End of story. Conservation triumph. Textbook example. Move on. By 1954, the combined population had grown to roughly 1,300 animals. Managers spent the following decades doing something that sounds almost absurd, given how recently the species had nearly disappeared. They began culling the herd, because bison reproduce efficiently, and the park's winter range could only support so many animals without triggering overgrazing and mass starvation. Thousands of animals were removed over the years, some given to Native American tribes and relief agencies. Some used to seed new conservation herds on other public and tribal lands across the country. Today, the population fluctuates between roughly 4,000 and 6,000 animals, and Yellowstone is celebrated, correctly, as home to the only plains bison population in the country that has lived continuously on the same landscape, as wildlife, without ever going through captivity or crossbreeding with domestic cattle. That last detail matters more than it sounds like it should, and it is the first thread that DNA analysis started to pull on. Almost every other bison herd in North America today, even herds that look completely wild and free-ranging, carry some trace of domestic cattle DNA in their genome. This is a legacy of the very rescue efforts that saved the species from extinction. Ranchers in the late 1800s and early 1900s trying to preserve bison any way they could, sometimes crossbred them with cattle, either intentionally to create hardier stock, or accidentally because bison and cattle were sometimes kept in adjoining or shared pastures. That cattle ancestry, even diluted many generations later, still shows up in genetic testing across a huge share of the continent's bison population, including many animals people assume are purely wild. Yellowstone's herd is one of the very few exceptions, a population that geneticists have repeatedly confirmed shows no detectable cattle introgression. In a sense, this is a herd of genetic ghosts, animals whose bloodlines trace back cleanly to the wild bison that survived the 19th century slaughter, untouched by the crossbreeding that shaped almost every other surviving population. But knowing the herd was pure was only the beginning. The real questions that scientists wanted answered went deeper. Did those two founding groups, the 23 wild survivors and the 21 introduced animals, actually merge into one true population, the way the simple textbook story suggested? Or did something more complicated happen, something the historical paperwork never captured? The first serious attempt to answer that question using modern genetic tools came from a study using nuclear microsatellites, a type of genetic marker scattered throughout an animal's chromosomes that can reveal patterns of relatedness and breeding structure within a population. Researchers analyzed dozens of these markers across the Yellowstone herd and found something that surprised a lot of people who assumed the population had fully blended a century earlier. The data showed evidence of population subdivision. In plain language, the bison appeared to still be functioning genetically as two separate breeding groups, even after 100 years of living inside the same park boundaries. One group tended to summer and breed in one part of the park, the other in a different area, and their genetic signatures were distinct enough that researchers could statistically detect the split. This was a strange result to sit with. These were not two herds separated by mountain ranges or hundreds of miles. These were bison sharing the same relatively contained landscape, moving across overlapping seasonal ranges, and yet the mathematics of their DNA suggested they were, in some meaningful sense, still behaving like the descendants of two separate founding populations from more than a century earlier. It raised an uncomfortable possibility. Maybe the neat story of total genetic merger that had been repeated for decades was wrong, or at least incomplete. Maybe those original bloodlines, the wild Pelican Valley survivors and the introduced Montana and Texas animals, had never fully blended the way everyone assumed. Then, in 2016, a team of researchers led by scientists at Texas A&M went even deeper, sequencing mitochondrial genomes, the small loop of DNA passed down exclusively through the maternal line, essentially an unbroken genetic thread connecting every animal back through its mother's mother, and so on, generation after generation. Mitochondrial DNA is prized in genetics precisely because it does not get shuffled and recombined the way most of an animal's DNA does. It is a comparatively clean record of maternal ancestry, and it can preserve historical signals that other kinds of genetic markers blur or erase over time. The team sampled 25 bison at random from across the Yellowstone population and identified 10 distinct mitochondrial haplotypes, essentially 10 distinguishable maternal lineage signatures circulating within the herd. Then they did something clever. They compared those 10 Yellowstone haplotypes against a broader set of 65 bison sampled from populations across North America, encompassing 30 different haplotypes total, built from more than 100 genetic marker positions. What emerged from that comparison was the finding that would eventually rewrite the understanding of this herd's history. The 10 haplotypes found in Yellowstone bison split cleanly into two separate genetic clades, two distinct maternal lineage groups, and the genetic distance between those two clades was large. Not a subtle statistical whisper, but a clear, structured signal, the kind of divergence you would expect from two genuinely separate historical populations rather than one continuous group. And when researchers traced those two clades back to their likely origins, the answer clicked into place with almost eerie precision. One lineage traced directly back to the 22 indigenous bison that had survived in central Yellowstone by 1902. The other lineage traced back to the 18 females introduced from northern Montana that same year. More than a century of shared range, shared seasons, shared winters and summers, millions of bison born and raised inside the same park boundaries, and the maternal genetic signal from those two founding groups was still cleanly detectable, still splitting the population into two identifiable historical branches. The mothers of 1902 were still, in a very real genetic sense, present and legible inside the mitochondria of animals grazing in Yellowstone more than 100 years later. Here's the detail that genuinely surprised researchers and forced a rethink of assumptions. When they mapped where these two lineages actually lived within the park today, they did not find them neatly separated into two geographic zones, north versus south, one valley versus another. Instead, the two mitochondrial lineages were spread throughout the park, mixed together across different herds and ranges, with no clean geographic boundary between them. Statistically, when researchers tested for geographic population subdivision using this maternal DNA, they found essentially none. The two founding bloodlines had spread and mixed across the landscape freely. And yet, confusingly, the earlier nuclear microsatellite study using a different kind of genetic marker had detected real population structure, real evidence of two separate breeding groups. How could maternal lineage data show total geographic mixing, while other genetic markers showed a persistent split? This is exactly the kind of contradiction that makes scientists sit up and take the problem seriously, because it means at least one of the studies was capturing something the other was missing, or the underlying biology was more complicated than a simple story about two herds either merging or not merging. Researchers proposed a few explanations. Different types of genetic markers can behave differently depending on how fast they mutate, how they are inherited, and what parts of population structure they are sensitive to. It was also possible that the population truly did retain some breeding level structure, some tendency for animals from one lineage to preferentially breed within certain seasonal groups, even while individual animals and their mitochondrial signatures move freely across the physical landscape. In other words, the geography had blended even while pockets of behavioral or reproductive structure lingered underneath the surface, invisible unless you were looking at the right kind of genetic evidence. It is worth pausing on how strange this must have felt to the researchers running the microsatellite analysis in the first place. They were not looking at a species that had been artificially split by fences or transported to different continents. They were looking at animals that had shared the same park, the same roads, the same rivers, the same predators, for generation after generation. Wolves came and went from the ecosystem. Elk populations boomed and crashed. Winters buried the park in snow, and summers turned the same grasslands gold, and through all of it, these bison moved across largely overlapping terrain. And still, buried in their nuclear DNA, there was a signal old enough and strong enough to separate them into two statistically distinguishable groups. It suggested that whatever behavioral or reproductive habits had been set in motion back in 1902, when the wild survivors wintered in one valley and the newly arrived Montana and Texas animals were kept and bred in another, those habits had somehow calcified into something durable, something passed down through the generations almost like a tradition, even after the physical separation between the two groups had long since become porous. For years, that unresolved tension sat in the scientific literature. One type of DNA evidence suggesting the Yellowstone bison were essentially one unified population with deep historical roots, split down a maternal line. Another type of DNA evidence suggesting the herd still behaved functionally like two distinct breeding populations descended from the 1902 founding event. Wildlife managers had to make decisions about how to handle this population, how to think about its genetic health, how to manage culling and transfers to outside conservation herds, all while working with an incomplete and somewhat contradictory picture of whether this was truly one herd or, genetically speaking, still two herds sharing a park. That is where the most recent research comes in, and it is the piece of this story that has scientists rechecking history all over again. A team from Texas A&M's College of Veterinary Medicine and Biomedical Sciences, working under the same lead researcher who had helped uncover the earlier mitochondrial split, went back into the park with a much larger and more strategically designed sampling effort. Instead of 25 random animals, they analyzed roughly 282 DNA samples, deliberately collected from both of the major summer breeding groups and both of the major winter ranges inside the park, essentially targeting exactly the geographic and behavioral divisions that earlier studies had flagged as potentially significant. They also used a newer, more powerful set of genetic markers alongside the original microsatellite loci from the earlier study, giving them a much sharper tool for detecting population structure than researchers had a couple of decades earlier. What they found overturned the lingering assumption that Yellowstone's bison were still, in any meaningful genetic sense, two separate populations. The new analysis detected no significant genetic differences between the summer breeding groups and no meaningful separation between the winter ranges. Gene flow, meaning actual breeding and genetic mixing, was extensive across what had previously been treated as separate herds. The animals sampled from different seasonal ranges shared genetic markers at a level fully consistent with belonging to one single interbreeding population, not two. For the first time, the genetic data lined up cleanly across different types of markers. This herd, after more than a century of shared history, had finally become what the old textbook story always claimed it already was: a single, unified, freely interbreeding population. Researchers involved in the study framed this in a particularly striking way. The population had not simply been unified from the start, contrary to the simplified conservation narrative that had circulated for decades. It had also not remained permanently divided, contrary to what the earlier microsatellite and mitochondrial studies had suggested. Instead, the real history was more dynamic and more interesting than either simple story. Two genetically distinct founding lineages, the wild Pelican Valley survivors and the introduced Montana and Texas animals, had spent generations gradually merging through natural processes, gene flow, population growth, range expansion, and shifting migratory behavior, slowly erasing a genetic boundary that had persisted for the better part of a century. What scientists were watching, essentially in real time across generations of sampling, was a population caught mid-merger, only reaching full genetic unification sometime in the past few decades. This reframes the entire history of the herd in a way that goes beyond a simple correction of an old assumption. It means that for most of the 20th century, when biologists and park managers spoke confidently about Yellowstone's bison as one recovered population, they were, in a genetic sense, not entirely correct. The population was recovering in numbers, yes, climbing from those 23 survivors and 21 introduced animals up into the thousands. But underneath that numeric recovery, two distinct bloodlines were taking generations longer to actually blend at the genetic level than anyone realized at the time. The near-extinction event of 1902 did not just threaten the species with disappearing, it also imprinted a genetic split that outlived the crisis itself by well over 100 years, quietly persisting through population growth, culling programs, and decades of management decisions before finally dissolving. There is also a deeper layer to this story that connects to how fragile the herd's genetic foundation really was, and how remarkable it is that the population is as healthy as it is today. When researchers modeled the effective population size during the recovery period, the number that matters for understanding genetic bottlenecks, they arrived at estimates as low as somewhere between 7 and 25 individuals, contributing meaningfully to the gene pool during the critical recovery window. That is an almost impossibly narrow genetic bottleneck for a population that would go on to number in the thousands. Populations that pass through bottlenecks that severe typically show significant loss of genetic diversity, elevated inbreeding, and reduced fitness. Yet when geneticists measured actual heterozygosity, a standard measure of genetic diversity across the modern Yellowstone herd using microsatellite analysis, they found levels around 63%, respectably high for a population that funneled through such a narrow bottleneck. This herd, in other words, should by the numbers be far more genetically compromised than it actually is. Textbook population genetics would predict a population funneled through an effective size that small to show clear signs of inbreeding depression, reduced fertility, weaker calves, more vulnerability to disease, a gradual narrowing of the gene pool with every generation, until the population becomes fragile in ways that are invisible until a bad year exposes them all at once. Yellowstone's bison largely avoided that fate. Some of that may come down to sheer luck in which individual animals happen to survive, and which ones happen to be chosen for the 1902 rescue effort. Some of it may come down to the specific genetic makeup of the Pablo Allard and Goodnight source herds, which, despite being privately raised, still carried substantial diversity of their own. And some of it may simply reflect how much genetic material can be preserved even in a tiny founding population, provided that population is given enough time, enough space, and enough protection to grow without immediately collapsing again. Whatever the exact mixture of causes, the outcome is a population that survived not just a near extinction event, but the kind of genetic squeeze that wipes out diversity in most species, and it came out the other side healthier than the math alone would predict. Put all of these threads together and you start to see why this particular DNA story has scientists going back through the historical record instead of just filing the new results away as a routine update. First, the population's purity, its complete lack of detectable cattle DNA, sets it apart from nearly every other bison herd in North America, most of which quietly carry the genetic fingerprints of the very crossbreeding programs that helped rescue the species. Second, the The maternal mitochondrial lineages of the 1902 founding event, the wild survivors and the introduced Montana females, remained detectable and distinct in the DNA for over 100 years, a genetic echo of a single pivotal year that outlasted generations of animals, management regimes, and assumptions about the herd's history. Third, evidence that had once been read as a stable, permanent divide between two breeding groups within the park turns out to have been a temporary, if remarkably long-lived phase, one that has only just resolved into true genetic unity within the current research team's own working career. And fourth, underlying all of it is a bottleneck so severe, an effective founding population potentially as small as single digits into the 20s, that the herd's survival and relative genetic health today reads less like an inevitable recovery and more like a genuinely improbable outcome. None of this changes the basic outline of the story that most people already know, that Yellowstone bison were nearly wiped out, that a small number of survivors and a small number of introduced animals were all that stood between the population and extinction, and that through a mix of legal protection, deliberate management, and simple biological persistence, that population grew into the thousands of animals roaming the park today. What the DNA adds is texture, complexity, and a kind of humbling reminder that even a story scientists thought they understood in broad strokes can still hide structure and detail that only reveals itself once you have the right genetic tools and enough patience to keep sampling the same population, decade after decade, as it continues to change. It also raises questions that researchers are still actively working through. If the two founding lineages only just finished merging into a single interbreeding population within recent decades, what does that mean for how the herd should be managed going forward? For years, decisions about culling, transfers to other conservation herds, and even routine population counts were made under an assumption of either full unity or partial division that turned out to be, at different points in time, both true and not true simultaneously. Understanding that the population has now crossed a genuine threshold into full genetic unification gives wildlife managers a clearer, more accurate picture to base future decisions on, whether that involves maintaining genetic diversity across transfers to tribal lands and other conservation herds, or simply understanding how resilient this particular population truly is compared to bison herds elsewhere that carry cattle ancestry, or that never experienced this specific kind of dual founder history. This is not a purely academic question either. Yellowstone's bison have become one of the most important genetic reservoirs for the species anywhere on the continent, precisely because of their purity and their unusual founding history. Since the population regularly exceeds what the park's winter range can sustainably support, hundreds of animals have been transferred out over the years, some to other public lands, and a growing number in recent years directly to tribal nations working to restore bison to reservation lands across the country. Herds have been reestablished on tribal lands in Montana, Kansas, Wisconsin, Alaska, and Iowa, among other places, using animals whose ancestry traces directly back through this same Yellowstone population, back through the two founding lineages of 1902, and further back still to the wild herds that once moved freely across the entire continent before the 19th century collapse. Every one of those transferred animals carries a piece of the same genetic history described here, which means the story of this DNA discovery is not confined to the boundaries of the park. It ripples outward into dozens of other herds and communities that depend on Yellowstone bison genetics to rebuild populations of their own. Knowing, with real confidence, whether the source population is one unified gene pool or a patchwork of semi-distinct lineages directly affects how conservation geneticists plan those transfers, how they think about long-term genetic diversity, and how they avoid inadvertently concentrating or diluting specific bloodlines as bison spread back out across parts of their historic range. There is something almost poetic about the fact that the final piece of evidence, confirming full genetic unification, came from the same broad research lineage, built on the same core genetic markers, refined and expanded over multiple studies spanning close to 2 decades, that had originally revealed the split in the first place. Science rarely resolves its own open questions in a single dramatic breakthrough. More often it happens exactly the way it happened here, in layers, one study raising a puzzle, a follow-up study offering a partial and sometimes contradictory answer, and eventually, with better tools and more patience, a clearer picture emerging that reconciles the earlier confusion rather than simply discarding it. What makes this particular case stand out is the sheer scale of history compressed into a strand of DNA. A crisis that played out in the span of a few brutal decades in the late 1800s, a rescue effort assembled in a single pivotal year, and then more than a century of quiet, ongoing biological negotiation between two bloodlines slowly working their way toward genetic unity, all of it recorded, waiting to be read. In blood samples drawn from animals grazing across the same valleys their great-great-grandparents once wandered as the last wild bison left, standing in the lower 48 states. Every one of those, roughly 282 samples analyzed in the most recent study, carried within it a trace of 1902, a trace of Pelican Valley, a trace of the Pablo Allard herd in Montana, and the Goodnight Ranch in Texas. Still legible, still readable, still telling a story that the written historical record alone never fully captured. Scientists rechecking history because of a DNA discovery is not really about proving old accounts wrong in some dramatic sense. It is about discovering that the full story was always more layered than the version that got passed down, and that biology itself has been quietly keeping its own parallel record the entire time, waiting for someone with the right sequencing tools to come along and read it properly. In the case of Yellowstone's bison, that record shows a herd that came within a hair's breadth of disappearing, that was stitched back together from a startlingly small handful of survivors and outsiders, that carried an invisible genetic seam between those two founding groups for well over a century, and that has only now, within the lifetime of researchers currently studying it, finally healed that seam into one true, unified, genetically resilient population. The animals grazing across Yellowstone's valleys today look to any visitor watching from a safe distance exactly like bison have always looked, but inside them is a genetic story still being actively rewritten, one sample at a time, and the next chapter of that story is very likely still being written right now out on that same landscape, in herds that carry within their cells the entire improbable history of how this species refused to disappear. Somewhere out in Lamar Valley or Pelican Valley right now, a bison calf is being born that carries, mixed together in a single genome, the maternal line of a wild survivor from 1902 and the maternal line of an animal shipped in from a Montana ranch that same year. 120 years ago, those two lineages belonged to animals that had likely never stood in the same field. Today, they belong to the same calf, standing on the same ground, part of the same fully unified herd. That is the quiet, almost invisible ending to a story that began with 23 animals and a species on the brink. And it is a reminder that sometimes the most dramatic history is not found in old photographs or written records at all, but in the double helix carried inside a living, breathing animal, still grazing, still surviving, still writing its own past into its future.